Hello everyone, welcome to a new session on the paper Cell Biology. In this module, we will study about the fine structure of chromosomes, prokaryotic and eukaryotic chromosomes with an emphasis on the special types of chromosomes such as Polytene and Lamb-Rush chromosomes. A chromosome is an organized structure of DNA and protein that is found in cells. A chromosome is a single piece of coiled DNA containing many genes, regulatory elements and other nucleotide sequences. Chromosomes also contain DNA bound proteins which serve to package the DNA and control its functions. The word chromosome comes from the Greek chroma, color, and soma, body, due to their property of being very strongly stained by particular dyes. They vary widely between different organisms. The DNA molecule may be circular or linear and can be composed of 10,000 to 1 billion nucleotides in a long chain. Typically, eukaryotic cells, that is, the cells with nuclei, have large linear chromosomes and prokaryotic cells. Cells without defined nuclei have smaller circular chromosomes, although there are many exceptions to this rule. Today, we know that a chromosome contains a single molecule of DNA along with several kinds of proteins. A molecule of DNA in turn consists of thousands and thousands of subunits known as nucleotides joined to each other in very long chains. A single molecule of DNA within a chromosome may be as long as 8.5 centimeters. To fit within a chromosome, the DNA molecule has to be twisted and folded into a very complex shape. Each chromosome has a constriction point called the centromere, which divides the chromosome into two sections or arms. The short arm of the chromosome is labeled the P arm. The long arm of the chromosome is labeled the Q arm. The location of centromere on each chromosome gives the chromosome its characteristic shape and can be used to help describe the location of specific genes. The arrangement of packets of genetic information in a chromosome is as described in the image. Furthermore, cells may contain more than one type of chromosome. For example, mitochondria in most eukaryotes and chloroplasts in plants have their own small chromosomes. The following are the different types of chromosomes. Prokaryotic chromosomes. The prokaryotes usually consist of a single giant and circular chromosome in each of their nucleoids. Each prokaryotic chromosome consists of a single circular double-stranded DNA molecule, but has no protein and RNA around the DNA molecule like eukaryotes. Different prokaryotic species have different sizes of chromosomes. Eukaryotic chromosomes. The eukaryotic chromosomes differ from the prokaryotic chromosomes in morphology, chemical composition and molecular structure. The eukaryotes, that is, plants and animals, usually contain much more genetic information than the viruses and prokaryotes, therefore contain a greater amount of genetic material. DNA molecule, which here may not occur as a single unit, but as many units called chromosomes. Different species of eukaryotes have different but always constant and characteristic number of chromosomes. In eukaryotes, nuclear chromosomes are packaged by proteins into a condensed structure called chromatin. This allows the very long DNA molecules to fit into the cell nucleus. The shape of the eukaryotic chromosomes is changeable from phase to phase in the continuous process of the cell growth and cell division. Chromosomes are the essential unit for cellular division and must be replicated, divided and passed successfully to their daughter cells so as to ensure the genetic diversity and survival of their progeny. They are thin, coiled, elastic, contractile, thread-like structures during the interphase and are called chromatin threads, which under low magnification look like a compact stainable mass, often called as chromatin substance or material. During metaphase stage of mitosis and prophase of meiosis, these chromatin threads become highly coiled and fold to form compact and individually distinct ribbon-shaped chromosomes. These chromosomes contain a clear zone called kinetochore or centromere along their length. Eukaryotes, that is, the cells with nuclei such as plants, yeasts and animals, possess multiple large linear chromosomes contained in the cell's nucleus. Each chromosome has one centromere with one or two arms projecting from the centromere, although under most circumstances these arms are not visible as such. In addition, most eukaryotes have a small circular mitochondrial genome, and some eukaryotes may have additional small circular or linear cytoplasmic chromosomes. Chemical structure of chromosomes. Chemically, the eukaryotic chromosomes are composed of deoxyribonucleic acid or DNA, ribonucleic acid or RNA, histone and non-histone proteins and certain metallic ions. The histone proteins have basic properties and have significant role in controlling or regulating the functions of chromosomal DNA. The non-histone proteins are mostly acidic and have been considered more important than histones as regulatory molecules. Some non-histone proteins also have enzymatic activities. The most important enzymatic proteins of chromosomes are phosphoproteins, DNA polymerase, RNA polymerase, DPN pyrophosphorylase and nucleoside triphosphatase. The metal ions as calcium and magnesium are supposed to maintain the organization of chromosomes intact. Molecular structure of chromosomes. According to the recent and widely accepted theory of Dupuy and Hans Riss called uni-stranded theory, each eukaryotic chromosome is composed of a single greatly elongated and highly folded nucleoprotein fiber of 100 angstrom thick. This nucleoprotein fiber in its turn is composed of a single linear double-stranded DNA molecule which remains wrapped in equal amounts of histone and non-histone proteins and variable amounts of different kinds of RNA. Dupuy produced a folded fiber model to show the ultrastructure of chromosome. Folded fiber model. Folded fiber model of chromosome structure was proposed by Dupuy in 1966, in which the chromosomal unit is regarded as a single DNA protein fiber which is repeatedly folded back on itself both longitudinally and transversely to make up the body of the chromatid. The model defines a unit chromatid as a chromatin fiber of variable length, of folding configuration and of replication mechanism. It corresponds to one continuous Watson-Crick DNA molecule. The single DNA helix of the unit chromatid is thought to be wrapped with polypeptides in P configurations, for example, lysine-rich histones, and then supercoiled to make up the 200 to 300 angstrom chromatin fibers. Presumably, supercoiling is induced and maintained by cross-linking with the helical proteins such as arginine-rich histones. During interphase S period, the unit chromatid replicates sequentially at 2 or more replication forks, a process that involves local unwinding of the tertiary and secondary helices. Each pair of daughter fibers corresponds to a pair of metaphase chromatids, which are held together by unreplicated portions of the fibre, especially in the centromere region. Nucleosomes and solenoid model of chromatin. In eukaryotes, DNA is tightly bound to an equal mass of histones which serve to form a repeating array of DNA protein particles called nucleosomes. If it was stretched out, the DNA double helix in each human chromosome would span the cell nucleus thousands of times. Histones play a crucial role in packing this very long DNA molecule in an orderly way into nucleus only a few micrometers in diameter. Thus, nucleosomes are the fundamental packing unit particles of the chromatin and give chromatin a beads on a string appearance in electron micrographs taken after treatments that unfold higher order packing. The nucleosome beads can be removed from long DNA string by digestion with enzymes that degrade DNA such as bacterial enzyme micrococcal nuclease. After digestion for a short period with micrococcal nuclease, only the DNA between nucleosome beads is degraded. The rest is protected from digestion and remains as double-stranded DNA fragments 146 nucleotide pairs long, bound to a specific complex of 8 nucleosome histones, that is the histone octamer. Kinds of chromosomes. The eukaryotic chromosomes have been classified into autosomes and sex chromosomes. The autosomes have nothing to do with the determination of sex and exceed in the number than sex chromosomes. The sex chromosomes determines the sex of the bearer. They are usually 2 in number and are usually of two kinds, X chromosomes and Y chromosomes. Special types of chromosomes. The eukaryotes, besides possessing the usual type of chromosomes in their body cells, contain some unusual and special types of chromosomes in some body cells or at some particular stage of their life cycle. The special type of eukaryotic chromosomes are Polytene chromosomes. Giant chromosomes were first observed by E.G. Balbiani in the year 1881 in nuclei of certain secretory cells, that is the salivary glands of chironomous larvae of the Diptera family. However, he could not conclude them to be chromosomes. They were conclusively reported for the first time in insect cells of Drosophila by Theophilus Painter of the University of Texas in the year 1933. Since they were discovered in the salivary gland cells of insects, they were termed as salivary gland chromosomes. Polytene chromosomes are oversized chromosomes which have developed from standard chromosomes and are commonly found in the salivary glands of Drosophila melanogaster. Specialized cells undergo repeated rounds of DNA replication without cell division or endomitosis to increase cell volume, forming a giant polytene chromosome. Polytene chromosomes form when multiple rounds of replication produce many sister chromatids that remain synapsed together. Polytene chromosomes are giant chromosomes common to many dipteran or two-winged flies. They begin as normal chromosomes, but through repeated rounds of DNA replication without any cell division or endoreplication, they become large banded chromosomes. The centromeric regions of the chromosomes do not endareplicate very well. As a result, the centromeres of all the chromosomes bundle together in a mass called the chromocenter. Polytene chromosomes are usually found in the larvae, where it is believed these many replicated chromosomes allow for much faster larval growth than if the cells remain diploid. Simply because each cell now has many copies of each gene, it can transcribe at a much higher rate than with only 2 copies in diploid cells. When stained and viewed under compound microscope at 40x magnification, they display about 5000 bands. In them, the chromomere or the more tightly coiled regions alternate with regions where the DNA fibers are folded loosely. A series of dark transverse bands alternates with clear zones of interbands. Such individual bands can be correlated with particular genes. About 85% of the DNA in Polytene chromosomes is in bands and rest 15% is in interbands. The cross-banding pattern of each Polytene chromosome is a constant characteristic within a species and helps in chromosome mapping during cytogenetic studies. In Drosophila melanogaster, there are about 5000 bands and 5000 interbands per genome. These chromosomes are not inert cellular objects, but dynamic structures in which certain regions becomes puffed out due to active DNA transcription at particular stages of development. These chromosome puffs are also termed Balbiani rings. Puffs may appear and disappear depending on the production of specific proteins, which needs to be secreted in large amounts in the larval saliva. Another peculiarity of the Polytene chromosomes is that the paternal and maternal chromosomes remain associated side by side, and the phenomenon is termed somatic pairing. Lamb-brush chromosomes. Lamb-brush chromosomes were first observed by Fleming in 1882 in sections of salamander oocytes and later described by Ruckett in the year 1892. They appeared like brushes used for cleaning lambs, hence the name lamb-brush chromosome. They are transitory structures and can be observed during the diploidine stage of prophase 1 in meiosis in the oocytes of all animal species, both vertebrates and invertebrates. They have been described in sepia, Icanaster, and in several species of insects, shark, amphibians, reptiles, birds and mammals. Lamb-brush chromosomes have also been found in spermatocytes of several species, giant nucleus of Acetabularia, and even in plants. Generally, they are smaller in invertebrates than vertebrates. They are observed in oocytes because oocytes are high in DNA content. Lamb-brush chromosomes are functional for studying chromosome organization and genome function during meiotic prophase. Additionally, Lamb-brush chromosomes are widely used for construction of detailed cytological maps of individual chromosomes. They are of exceptionally large sizes and present in bivalent form. They are formed due to the active synthesis of mRNA molecules for future use by the egg cells when no synthesis of mRNA molecule is possible during the mitotic cell division. Lamb-brush chromosomes are clearly visible in the light microscope. They are organized into a series of chromomeres with large chromatin symmetrical loops extending laterally. Each loop appears at a constant position in the chromosome, that is 10,000 loops per chromosome set or haploid set. Each loop has an axis made up of DNA unfolded from the chromosome and is transcriptionally highly active, wherein several transcription units with polarized RNP matrix coats the DNA axis of the loop. The majority of the DNA, however, is not in loops but remains highly condensed in the chromomeres on the axis and lacks expression of genes. Let us now summarize what we have learnt in this particular session. Eukaryotic chromosome structure refers to the levels of packaging from the raw DNA molecules to the chromosomal structures seen during metaphase in mitosis or meiosis. Chromosomes contain long strands of DNA containing genetic information. Compared to prokaryotic chromosomes, Eukaryotic chromosomes are much larger in size and are linear chromosomes. Eukaryotic chromosomes are also stored in the nucleus of the cell, while chromosomes of prokaryotic cells are not stored in a nucleus. Each chromosome consists enormously long linear DNA molecule associated with proteins that fold and pack the fine thread of DNA into a more compact structure. The nucleosome consists of a DNA double helix bound to an octamer of core histones, that is 2 dimers of H2A and H2B and H3 or H4 tetramer. About 147 base pairs of DNA coil around one octamer and 20 base pairs are sequestered by the addition of the linker histone or H1 and various length of linker DNA, that is 0 to 100 base pair, separate the nucleosomes. Lamb-Rush and Polytene chromosomes are two important special types of chromosomes. Since they were discovered in the salivary glands of insects, they were termed as salivary gland chromosomes. Polytene chromosomes are oversized chromosomes which have developed from standard chromosomes and are commonly found in the salivary glands of Drosophila melanogaster. Lamb-Rush chromosomes have also been found in spermatocytes of several species, giant nucleus of Acetabularia and even in plants. Here are a few questions for you to work out. 1. Describe the fine structure of chromosome. 2. Describe the folded fiber model of chromosome. 3. Differentiate between eukaryotic and prokaryotic chromosome. 4. Briefly describe the structure of Polytene and Lamb-Rush chromosome. 5. Describe the solenoid model of chromosome structure. Here I can suggest a few books for your reference. The Cell, a Molecular Approach, written by Jeffrey M. Cooper, Robert E. Hausman, 3rd edition, ASM Press. Biology, written by Peter H. Raven, George B. Johnson, 5th edition, McGraw-Hill. Advanced Molecular Biology, written by R. M. Twyman, 3rd edition, Bioth Scientific Publishers. Thank you for watching this program. Let's meet in the next session with another topic. Till then, bye.